All right, so as I was saying earlier, we will close the docket on enzymes with the last topic, enzyme kinetics. Now, what is enzyme kinetics? What is it when we say enzyme kinetics? So you remember when we were looking at the factors that influence enzyme kinetics, we looked at factors such as enzyme concentration, substrate concentration, temperature, and pH. Now, in enzyme kinetic studies, what you do is that you want to see how changes in these parameters affect the rate of a reaction that is an enzyme catalyzed reaction. So that is an enzymatic reaction. You have an enzyme which is catalyzing a reaction, and you are changing these parameters to see how they influence the rate of the reaction. Of course, when you do that, you keep other parameters constant. So for instance, if you are studying the influence of enzyme concentration, you will then keep the other parameters like temperature, pH, substrate concentration constant, and vary only the enzyme concentration. So you do that for each of these parameters to see how they influence the rate of the reaction. So when you do that, all you are doing is you are studying kinetics of the reaction, and because it's an enzymatic reaction, you refer to it as enzyme kinetics. Indeed, the curves that we saw, when we were studying the factors that influence enzymatic reactions, and we're looking at substrate concentration or enzyme concentration, we're looking at certain curves, certain graphs. Those curves actually are kinetic curves. So those are kinetic curves from, say, studies of how those factors influence the enzymatic reactions. So that is basically what it means when we say enzyme kinetics. Now, a bit of some historical background. I mean, currently the accepted enzyme kinetics internationally, which is used, the simplest of the kinetic systems that are used in the studies of kinetics is what is referred to as the Michaelis-Menten kinetics. And we'll come to see the Michaelis-Menten equation, Michaelis-Menten constant. But a bit of historical background, first of all. So the Michaelis-Menten kinetics actually, started from, studies of Victor Henry. The picture on the left of your screen, that's Victor Henry. Victor Henry is actually a Russian, but he was born in France. So on paper, he's French, but he was born to Russian parents. Of course, there's some history behind all of that, why he had to be born in France and be a French on paper, whilst actually he's Russian. For that, I'll leave it out. I mean, those of you who are interested, you can go and read much more on Victor Henry, and you will see why he was a French on paper instead of, even though he has Russian parents. You can go and read on that, because that is not really part of the biochemistry. But if you want to know about the life of the man, you can go and read on it. So he actually was the first person who represented the activity of enzymes, or say the kinetics of enzymes, with some mathematical equation. So he did some studies and came up with an equation to represent the activity of enzymes, I mean, based on substrate concentration. But of course, at the time, many biochemists didn't understand the significance of the equation that he published. He published a paper in which there was that equation. Right? Over a decade later, before Leonard Michaelis and Leonora Menton were studying, his works, what he did earlier, before they came up with the Michaelis-Menten kinetics. And Leonard Michaelis he was a German. He was working as a professor in one of the universities, but later he had to leave, go and work in Japan for some time, and then later went to the United States. That's where he actually continued and retired in the United States. Now, Leonora, she was one of the first female medical doctors in Canada. But after she finished with her medical education and wanted to go into scientific research, at the time in Canada, women were not allowed to participate in scientific research. So she has some really motivational story there. For a bit, so I'm waiting a bit so that when it comes back, then I continue. Okay, so my network was broken a bit, so I stopped for some time so that after now it has come, then I can continue. All right, so Leonora traveled to Germany because she wanted to go into scientific research, because she couldn't do that in Canada. So that is a determined, woman. And so in Germany, she worked together with Leonard Michaelis on the Michaelis-Menten, the work they did before they came up with the Michaelis-Menten kinetics and then the Michaelis-Menten equation. And that has become the fundamental, principle, fundamental system of enzyme kinetics up to today. Of course, there are other forms of enzyme kinetic studies, but we won't bother ourselves so much about that. We will just focus on the Michaelis-Menten because this is what is accepted, all over now. So I don't want us to go into that. All right. So what did they do? They studied, the changes in substrate concentration, product concentration in an enzymatic reaction. So what you are looking at is a kinetic curve where you have concentration on the vertical axis, time, on the horizontal axis. And you see the curve for the substrates from the beginning. Substrate is high in concentration, and the substrates, it reduces all the way until all the substrate is consumed in the reaction system. And you have a product which at the beginning of the reaction is 0, and that product also begins to increase all the way up to the end of the reaction. I mean, these things are logical. That when you think of it, that at the beginning of a reaction, yes, you have a substrate that an enzyme should be acting on. That enzyme will be in high concentration, is not yet consumed. And as the substrate is being consumed to form products, then the concentration of the substrate will be reducing until all the substrate is consumed. Except that when you study, then you can see actually the curve, how the change is occurring over time. And the same way how the product also increases in concentration, from the beginning of the reaction all the way to the end of the reaction. Then there were some other two things, which was the concentration of the enzyme, which also at the beginning of the reaction is high, and it begins to reduce all the way to a certain point where you see it, barely being constant, and then coming back to the concentration, the initial concentration that it was. I mean, that is to say that the enzyme, we talked about this, that in an enzymatic reaction, the enzyme is not consumed. The enzyme is there as a catalyst. It comes in to speed up the rate of the reaction. And then after that, the enzyme will remain. It's not consumed in the process. And we also saw that in the process of enzymatic reactions, you have what we call the enzyme substrate complex, the transition state, the enzyme substrate complex, which goes to the transition state and then gets converted to the product. And we see also that from the beginning of the reaction, you have that enzyme substrate complex, being formed. It increases in concentration all the way and then comes down. Of course, the enzyme substrate complex also goes to transition states and gets converted to a product. So in the end, all the enzyme substrate complex will get consumed out of the system. So you will no longer have any enzyme substrate complex there. Now, if you compare what they did to Henry Victor, they considered some more other things, like the formation of the enzyme substrate complex and these concentrations of the enzyme and the enzyme substrate complex, the different stages of the reaction, which Victor Henry didn't consider. So we can look much more at these stages. And we see the very first stage of this reaction, where we see that there is the formation of the enzyme substrate complex. We see what we refer to as the pre-steady state. You see the enzyme concentration is up, whilst the enzyme substrate complex is low. So these concentrations are widely apart, and they begin to close up all the way until you see that the enzyme substrate complex which is formed is almost constant there. You see what, when we were looking at the influence of the concentration of substrates, we saw there was a saturation curve. So it's a similar thing you are seeing here, that at a certain point you have an enzyme substrate complex formed and the concentration is almost constant. It's not changing there. Of course, that is understood because you have a certain amount of enzymes, inside the reaction mixture. And it is that amount that binds to the amount of substrates which is present. And once all the enzyme which is present in the reaction mixture is bound to a substrate, there will not be any change in that enzyme substrate complex formed again, because all of it is saturated, so there will not be any further changes in the concentration of the enzyme substrate complex. Similarly, you have the concentration of the enzyme also being steady at that point, so also constant. And right after that, you see that the enzyme substrate complex begins to go down all the way until all the enzyme substrate complex is consumed in the system. And once the enzyme substrate complex is getting converted to product, then of course you'll be having free enzyme molecules back into the system. So you have all these stages, the pre-steady state, the steady state, that have been considered in the Michaelis-Menten kinetics, which gives more perspectives into the kinetics of enzymes than the earlier study of Victor Henry. Now, with that, Michaelis and Menten were able to come up with an equation to represent the kinetics of enzymes, what we refer to as the Michaelis-Menten equation. Of course, sometimes it's been said that maybe the equation should be referred to as Henry Michaelis-Menten kinetics, because Michaelis and Menten actually built on the work of Henry, so that his efforts will also be his efforts will also be recognized as well. But anyway, sometimes in some books you may see it being said to be Henry Michaelis-Menten kinetics. Some refer to Henry Victor kinetics. Some will say Michaelis-Menten kinetics. But of course, the history is there, so we know who did what and who built on what. So, well, if later the decision is taken, maybe in it will become generally what will be accepted in books that will be seen Henry Michaelis-Menten. But now you will see diverse views. In some books, you will see it being written like that, Henry Michaelis-Menten kinetics. In some others, you will see only Michaelis-Menten kinetics. But you will also see the Henry Victor kinetics, his first study before the Michaelis-Menten kinetics was done. Now, what you are seeing is the saturation curve, which we saw much earlier when we were looking at the influence of substrate concentration on enzymatic reactions. And so you see the substrate concentration is on the horizontal axis and reaction rate on the vertical axis, for you to see the dependence of the rate of the reaction on substrate concentration. And just like, if you remember, your secondary school core mathematics, where you look at equation of a line. So if you take a tangent of this curve to say a straight line from 0 going up, you can find, equation of the line as an estimate. And if the equation of that line being what y equals ax plus b, y being the value which is on the y-axis, and A is the gradient, X, of course, the value on the X-axis. B, that is the intersection on the Y-axis. But the intersection here, of course, because it's the concentration is 0 from the beginning. So there, the intersection is going to be 0. So when that equation is sort of derived, you come to what we have as the Michaelis-Menten kinetics, the Michaelis-Menten equation, where you have the V or V null, which is the rate of the reaction, equals the maximum rate of reaction times concentration of the substrate divided by the Michaelis-Menten constant, which is written as Km, plus concentration of the substrate. Now, what you pay attention to on this curve is that the Michaelis-Menten constant is shown as the concentration of the substrate at which half of the maximum of the rate of the reaction is reached. So that is the Michaelis-Menten constant. See, the maximum rate of the reaction is that, the maximum rate that that reaction can be reached, because you see the curve plateaus. So whatever you do, even if you are adding more substrate concentrations, more substrate molecules into that enzyme, that enzymatic reaction mixture, the rate of the reaction does not increase. We saw this already, this saturation curve already. And so you have a maximum rate of the reaction over there. To reach half of that, you need a certain amount of substrate, a certain concentration of the substrate. And that's concentration of the substrate at which you reach half of the maximum of the rate of reaction is what is referred to as the Michaelis-Menten constant. Now, this value is significant in the sense that it shows affinity of an enzyme to a substrate. What do I mean by that? So if you have, let's say if you are studying two enzymes and you have one of the enzymes having a curve, similar to this, except that you have the Michaelis-Menten constant being greater. That is the amount of substrate that is needed to reach half of the maximum of the reaction is higher. It means that enzyme has less affinity. In other words, the more or the greater the Michaelis-Menten constant, the lesser affinity of an enzyme to a substrate. Another way, the lesser the Michaelis-Menten constant, the higher the affinity of an enzyme for a substrate. If you think of it, it's also logical that if an enzyme has more affinity to a substrate, then of course you will need lesser amount of the substrate for the rate of the reaction to be, to increase, to go up all the way up to half of the maximum. But if you have the enzyme having lesser affinity for the substrate, then you need more substrate to reach half of the maximum of the reaction. So that is the significance of the Michaelis-Menten constant. All right. Now, with that foundation laid, we can move on to take a look at what we refer to as enzyme inhibition, and then we can look at some practical applications of enzyme inhibition. In fact, in the pharmaceutical sector, lots of drug discovery is based on enzyme inhibition. So we'll take some specific examples, even in the development of things like pesticides, for instance. You also have some applications of enzyme inhibition in this aspect. Of course, there's a whole lot that can be said about enzyme inhibition, application of it as in the discovery of pharmaceutical agents and in discovery of say pesticides and things like that. But of course, in this short lecture, 1 hour, 2 hours, you cannot cover all of these things. But as much as possible, we'll do some simple things that I hope by the end of the day it will sort of whet your appetite such that you go, maybe you go to a pharmacy, you buy a medicine, or even those of you working in the pharmacies, or maybe working in some hospital or anything. You take a medicine, you look at the leaflets, and when you are reading, you have more understanding of how the medicine actually works. And you pay attention to certain enzymes that are mentioned, particularly when you are looking at the portion where it's talking about pharmacokinetics. You have more interest in looking at okay, this is the mechanism by which the medicine works. Or maybe if you take a pesticide and you look on the container and look at the active ingredients, you begin to understand how this pesticide is acting as an inhibitor to a certain enzyme, which will give a certain effect in an insect, for instance, for you to be able to control them and things like that. So that in the end, I mean, that's what it's supposed to do. When you come to study these things, you're supposed to apply it in real life. It's not just to read them and write exams, pass, and things like that. So at least you have some better understanding of these things. Anytime you pick any of those leaflets or you pick any of those medicines, you begin to relate to the mechanism of action of these medicines, how they work, and how they achieve the effect that is intended. So we talked about coenzymes and cofactors as activators of enzymes. These are some prosthetic parts of enzymes, non-protein parts. They are required to make an enzyme active. We also have what can slow down the activity of an enzyme or even terminate the activity of an enzyme. These we refer to as inhibitors. Now, these inhibitors can serve as pharmaceutical agents, as I was saying that a lot of times drug discovery is based on enzyme inhibition. And you also will find the use of inhibitors just in research, enzymatic reaction mechanisms, the study of it. Of course, enzymatic reactions can be fast. If you are able to slow down the reaction, then of course you'll be able to study it much more, looking at maybe intermediates that are formed and all of that. Or just because you are just studying a particular reaction system that you want to control. That is, for instance, if you want to develop some medication for a particular disease condition, and you want to use a particular compound as an inhibitor, of course, you'll be using that in your study of the kinetics of the enzyme. So, yes, inhibitors are used a lot in research works, that is into enzyme kinetics. Right. So we'll take a look at some forms of inhibition. And the first one we're looking at is competitive inhibition. And as the term suggests, there will be some form of competition. So this is the red structure you see is depicting an enzyme with an active center in it. And in that active center, there is a substrate that is bound. That is the normal substrate. So you have a normal substrate that binds to the active center of an enzyme. Then you can have an inhibitor, which is a competitor to the normal substrate. So you see that inhibitor is having a similar structure to the normal substrate. So that is why it's a competitor. It looks like it's having, so sometimes we refer to it as a structural analog of the substrate. Structural. So it's having a structure that is similar, not having the same function. It's not a functional analog. It is having a structural similarity that can bind to the active center, but it will not have the same effect as a substrate. So you see, you have that inhibitor acting as a competitor to the substrate. And once it binds in there, it prevents the normal substrate from binding to the enzyme. That way, the inhibitor prevents the enzymatic reaction from occurring. So that's a competitive inhibition. Now that competitive inhibition, we can represent it with a Michaelis-Menten plot or the Michaelis-Menten kinetics, such that if you look at these two curves, the red curve represents a normal enzymatic reaction, uninhibited. There is no inhibitor in that reaction. That's the red curve. And we see the blue curve represents the kinetics of the inhibited reaction. So the blue curve is having a competitive inhibitor in there. And what do we see? What's the difference that we see here? We see that, well, half of the reaction rate, or half of the maximum rate of the reaction is not changed, it's the same. What it also means is that the maximum rate of the reaction has not changed, it's the same. Even though you can see that the maximum rate of reaction for the blue curve, to the red curve is low, it doesn't mean that the maximum rate of the reaction cannot be reached when you have a competitive inhibitor. We talked about this before, that you have a competitive inhibitor in the reaction mixture, because it is binding to the active center. If you introduce more substrate molecules into the system, if you increase the concentration of the substrate, you increase the probability of the substrate molecules binding to the active center. And actually, if you have a competitive inhibitor in a reaction mixture and you add more substrates into the reaction mixture, that substrate molecules, the extra substrate molecules which you are adding to increase the concentration of the substrate, will go to displace the inhibitor which is bound to the substrate, so that you can still reach the maximum rate of the reaction, only that it will take you a longer time to reach that because of the presence of the inhibitor. So you have half of the maximum rate of the reaction being the same, but you notice that the Michaelis-Menten constant has increased in the case of the inhibited reaction. Where you have the competitive inhibitor, you notice that the Michaelis-Menten constant has increased. The concentration of the substrate at which half of the maximum rate of the reaction is reached is more. Yes, it's more because you have a competitive inhibitor there which is also binding to the enzyme, and you need more substrate in the system in order for the reaction to reach that level of, the rate of the reaction. You can also look at it as if the competitive inhibitor is sort of reducing affinity, because we talked about affinity. So you can see it happening over here with the competitive inhibitor, such that when you compare the Michaelis-Menten constant of the uninhibited reaction, and then you have, this usually will be called an apparent Michaelis-Menten constant in the inhibited reaction. You see, the apparent Michaelis-Menten constant is greater than the Michaelis-Menten constant for the uninhibited reaction. Then we also have what we refer to as non-competitive inhibition. In this instance, you have the substrate binding to the active center, all right, but you have the inhibitor binding to not the active center, but to another site on the enzyme, usually referred to as an allosteric site, where you have the inhibitor binding to that enzyme substrate complex. And the binding of the inhibitor to the enzyme substrate complex prevents that enzyme substrate complex from proceeding in a reaction, getting to the transition state and getting converted into a product. Of course, when you look in biochemistry books, sometimes even this type of inhibition is also divided into different forms. We just don't want to so much go into some of these things, but I can mention them later. If you yourself you are interested, you can go and look at it. Sometimes you have also what is referred to as uncompetitive inhibition, where you have the inhibitor binding first to the enzyme to form an enzyme inhibitor complex before the substrate comes to bind. In this case, you see that the substrate binds first to form an enzyme substrate complex before the inhibitor comes to bind, which is being referred to as a non-competitive inhibition. Of course, some of these things are theoretical. And then you see systems that are modeled to show that you can have what we refer to as mixed inhibition, where you have both non-competitive and uncompetitive inhibition, things like that. But we will not be going so much into these things. This time, we just want to refer to competitive inhibition, non-competitive inhibition, just to understand the principle by which these things occur. And that too, we can represent it with a Michaelis-Menten curve. In this instance, what you notice is that the Michaelis-Menten constant hasn't changed, it's the same. So you see the concentration at which you reach half of the maximum rate of reaction is the same. But the maximum rate of the reaction has changed. So has half of the maximum rate of the reaction changed. In the sense that where you have the inhibitor, the non-competitive inhibitor, you have half of the maximum rate of the reaction lower than what you have for the uninhibited. Of course, because the inhibitor is not competing with a substrate. So whether the substrate goes to bind to the active center or not, that allosteric site is still present for the inhibitor to bind. And once it binds there, it's going to prevent the reaction from proceeding, or it will slow down that reaction so that whatever it is, you will not be able to reach the maximum rate of the reaction. So the maximum rate of the reaction reduces significantly. The same amount of substrates, which is giving you half of the rate of the reaction, half of the maximum rate of the reaction for the uninhibited, is giving you half of the maximum of the rate, half of the maximum rate of the reaction for the inhibited reaction, except that these rates, the rate of the inhibited reaction is lower, even though you are having the same amount of substrate in there. So that is the difference that you have between competitive and non-competitive inhibition. We can also classify enzyme inhibition in two ways, saying is either it is reversible or irreversible inhibition. So you have inhibitors that bind to enzymes. So what you see being depicted here is that you have a protein target that's the enzyme. The protein target is the enzyme, and the inhibitor is what is being shown as orange. And that inhibitor can bind to the enzyme and can also dissociate away from the enzyme. So the that inhibition process is reversible. So you find this with some drugs. I mean, I think we saw the example of succinylcholine, for instance, that we saw as one of the practical use of in enzymes in simple surgeries, where succinylcholine binds to acetylcholine esterase, and because of that, it's slowly dissociates away, so that you can have the muscle relaxed. So succinylcholine being used as a muscle relaxant, but in the end, it dissociates. So that is reversible. But you can also have irreversible inhibition, where the inhibitor actually forms covalent bonds with, or let's say strong non-covalent bonds also can be formed with the enzyme. That this is what you find maybe in pesticides, where you have some covalent bonds of inhibitors with a particular enzyme, that will result into, the death of an insect, for instance. Or maybe you're referring to a bactericidal antibiotic. The effect it will be having is irreversible, for instance. So you can have reversible, irreversible inhibition as well. Now, we want to take a look at some specific examples as a way of activating our interests into how this enzyme kinetics or enzyme inhibition is applied in drug discovery, development of drugs and things like that. So let's start with the system of the synthesis of tetrahydrofolate. Now, tetrahydrofolate is one of the coenzymes that we saw. We refer to it as also, called folic acid. And you remember we talked of the tetrahydrofolate as, that substance which is required in cell proliferation, the growth of cells, in cell specialization, and because of that, it's needed even for the growth of the fetus. You remember all of that. The same way, you have the tetrahydrofolate also required by bacteria in bacterial cell growth, for instance. So the same, you have such system of the synthesis of tetrahydrofolate. Now, we are seeing the process of the synthesis of tetrahydrofolate in 3 reactions. So if you look at it, 3 arrows. See how I tell you that when you just take 1 arrow at a time, if you see the arrows 1, 2, 3, it's 3 reactions. And we see you have a first reaction where you have dihydroterine pyrophosphate reacting with 4-aminobenzoic acid to give you dihydroteric acid. Really, what is happening? You are just having a conjugation of these 2. I mean, it's even convenient that we can see this part is red, and we see the 4-aminobenzoic acid is blue. So you can see that the 2 molecules just joined. What is referred to as a conjugation reaction. So that's a conjugation. And you have a dihydroteric acid. Now, you see, just that these 2 phosphate groups are gone. It's a pyrophosphate or a diphosphate. It's almost like that's an activation of that molecule to go into a reaction. And you have 1 hydrogen from that, primary amine group also gone. And then you just have that bond there. And you are having a conjugation of the 2 molecules. All right. The next thing you see is that you have an amino acid. This is green. Glutamic acid or glutamate coming to bind to give you dihydrofolic acid. And then the next thing you see, what is the difference between this dihydrofolic acid and this tetrahydrofolic acid? You see the enzyme that is there is dihydrofolate reductase. So there must be some reduction reaction that has occurred. And where has the reduction occurred? So if you look at the dihydrofolic acid, you look at the second ring. You know the two fused rings, the second ring, the red ring. You see that nitrogen there? That nitrogen is not having any hydrogen on it. But when you come down, you see that nitrogen is having a hydrogen on it. So that is where the reduction has occurred. So there's a reduction reaction. So that's how come you are having a tetrahydrofolic acid. All right. So we see this process of the synthesis of, tetrahydrofolate, or you can just say folic acid. Now, what we're going to do is we are going to zoom in on the first reaction and the last reaction. We are going to, you see, I tell you, take one arrow at a time. So we are going to take the first arrow and then we'll take also the last arrow. So we take the first arrow where you have the dihydroterine pyrophosphate reacting with 4-aminobenzoic acid. And 4-aminobenzoic acid is also referred to as para-aminobenzoic acid. Para only means opposite. So you have the carboxylic acid group and the amine group. They are opposite each other. So that is para. So that anywhere you put the carboxylic acid, you put the amine group opposite it, it will still be giving you 4-aminobenzoic acid, because that carbon on which the carboxylic acid group is becomes carbon number 1. And if you count 2, 3, 4, you see you have the amine group on the 4th carbon. So that's why I have 4-aminobenzoic acid, but it's the same as para-aminobenzoic acid. And so that first reaction that we saw, you have a conjugation to give you dihydroteric acid. Now, there's a small note that is written beside the arrow, which is sulfonamides. Sulfonamides. Now, sulfonamides is a class of antibiotics. It's a class of antibiotics. And so you see there's a cross on that arrow showing that sulfonamides act as inhibitors of that reaction. And specifically, sulfonamides act as inhibitors to this enzyme, dihydroteroid synthetase, because that is the enzyme which is catalyzing the reaction. Now, we see here the enzyme. What is the substrate that sulfonamide is competing with? Because sulfonamide is a competitive inhibitor. We see at the top, irreversible competitive inhibition. So what is the substrate? The substrate is 4-aminobenzoic acid or para-aminobenzoic acid. And we said competitive inhibitors are structural analogs. They look like the substrate. And so it's sort of the inhibitor is deceiving the enzyme, as if it's having the same structure and then it binds to the enzyme, but because it's not having the same function as the substrate, once it binds, the reaction does not proceed. So we see the structure of a sulfonamide put side by side with para-aminobenzoic acid for us to see the similarity. And we see both of them are primary amines. That is, you have amine group on the ring, except that in place of the carboxylic acid group in the para-aminobenzoic acid, you are having some sulfoamide group in place of that. Of course, not so the same, but there is that similarity between them. Of course, that is going to also go into what we refer to as specificity of these antibiotics. We have this like a broad spectrum antibiotics that's not so specific, but will be acting on a class of bacteria or classes of bacteria, things like that. Now let's go to the next one. We are now taking the third reaction, the last reaction. And that last reaction, that was where we saw the reduction of dihydrofolic acid, where you saw the nitrogen of the second ring of the dihydrofolic acid, now having a hydrogen on it in tetrahydrofolic acid. So it's a reduction reaction, which is catalyzed by dihydrofolate reductase. And here too, beside the arrow, there's a small note, trimethoprim. So trimethoprim is also an antibiotic, which is also acting as an inhibitor of this reaction, an inhibitor to which enzyme? The dihydrofolate reductase. So we see what is the substrate in this case. Of course, the substrate is this dihydrofolic acid, which is getting reduced to tetrahydrofolic acid. And if you look at the structure of trimethoprim, also not so much similar, except you can see the first ring. There's some amine group there. The nitrogens are there for the first ring, but in place of that double bond O, for the dihydrofolic acid, you have another amine group there. And there's even a CH2 between the two rings, but it also acts like a structural analog of this dihydrofolic acid and acts as an inhibitor to dihydrofolate reductase. So in that case, trimethoprim is used also as an antibiotic. Sometimes you see some urinary tract infections and things like that. Okay, so that is also a real life example there. We can take a look at another one. The same reaction, the same third reaction. This time around, the inhibitor, the drug there is methotrexate. Methotrexate. And methotrexate is, I think, it's used in chemotherapy against cancers. And we see it's also acting as an inhibitor to that enzyme. If you take a look at the structure of methotrexate, you can see that it's looking much more similar than the examples that we've seen earlier. Because if you look at the first ring, it's only that there's a double bond. There's a double bond O for the dihydrofolic acid on that first ring. In place of it, there is an NH2. And then if you look at these two nitrogens in the rings, of the dihydrofolic acid, they are having hydrogen on them. But if you come down to methotrexate, these nitrogens are not having hydrogen on them. And they have a double bond there because the hydrogens are gone, so they now have a double bond forming there. So you can look at the methotrexate in terms of this ring as some oxidized form. But of course, if you look at also the fact that you have NH2 here, so there's both some oxidation, reduction occurring on different groups. And you can also see on this nitrogen in methotrexate, this nitrogen, see there's a methyl group, CH3, CH3. But in dihydrofolic acid, it's hydrogen. There is no CH3 there. I think all other things are the same for the molecule. If you compare the two, those are the only differences between them. But this molecule methotrexate look very, very, very much like dihydrofolic acid. And that, of course, will make methotrexate much, much, much more specific to the cancer cells, so that you reduce adverse effects, things like that. It goes in there to prevent the synthesis of folic acid by the cancer cells. And once folic acid is not being synthesized, you don't have the growth of the cancer cells, and so the growth of tumor is minimized, so that in the end you have that intended effect of reduction of the growth of tumors. But of course, can also have some side effect because once it's targeting synthesis of tetrahydrofolic acid, it means generally synthesis of tetrahydrofolic acid in the body will go to affect other things, which will result into maybe the loss of hair, for instance, or some scaling of the skin, because it goes to affect, the growth of cells in general. And so you begin to have some of these effects which occur with chemotherapy. Right. Let's take another one. So we've seen the structure of peptidoglycan. We talked about peptidoglycan as the major component of the cell wall of bacteria. So if you look at this structure, this is a bacteria. Let's say that's you can call it a lactobacillus. Let's just say it's a bacillus. And we zoom in on the membrane there, the outer covering of it. And that is, made up of some phospholipid bilayer, that is the plasma membrane, which is protected by what we refer to as the peptidoglycan layers. And the peptidoglycan layers, too, you see there are some key here that we can look at to see what these things mean. So the pink spheres, the pink circles there, are N-acetylglucosamine, molecules, and the blue are N-acetylmuramic acid. I think you remember when we were talking about lysosomes. We referred to the fact that bacterial cell walls are made up of peptidoglycan, and lysosomes, go in there to break, glycosidic bonds between N-acetylglucosamine and acetylmuramic acid. Remember that. So you can quickly refer to that, and you see that aside the fact that you are having this polysaccharide, made up of N-acetylglucosamine and acetylmuramic acid. You also have what is referred to as some peptide linkages, so which are shown here between the polysaccharides, which is what makes the whole structure a peptidoglycan. The glycan is the carbohydrate part, but the cross-link between them is made up of peptides. So you are having some two peptides that are having a cross-linkage between N-acetylmuramic acid residues. So that is the structure of the peptidoglycan, which is a major component of the cell wall of gram-positive bacteria. Okay. Now, let's take a look at the biosynthesis of the peptidoglycan, particularly to the formation of the peptide linkages between the glycan molecules, the polysaccharide molecules. So we see that the glycan chain is represented with these bars, these teal and green bars, so that the teal color refers to N-acetylglucosamine and the green refers to N-acetylmuramic acid. And so you see the alternation. You have N-acetylglucosamine, then the green comes N-acetylmuramic acid. Then you have N-acetylglucosamine, you have N-acetylmuramic acid in that order. So you have that chain. And you also see that you have on the N-acetylmuramic acid, some peptides. So you have peptides that are on the N-acetylmuramic acid residues in that chain. Now you also see what is referred to as penicillin binding protein. Actually, if you are talking about the activity or the function of this protein, it is a glycopeptide transpeptidase, because it is that protein which is responsible for the formation of the cross linkages between the peptides that are on the N-acetylmuramic acid residues in the glycan chain. But if you think of it as that is the protein that is targeted by the antibiotic penicillin, or let's say penicillin family of antibiotics, then is being referred to as penicillin binding protein. And you see that in the active center of that enzyme, you have what is being shown as a reactive serine. So here again, you are seeing, so you can go back to the mechanism of action of enzymes, where we were referring to some serine proteases. So enzymes having serine in their active centers, and then you'll be seeing things like a proton transfer. We saw these things, so you can see that this protein is also one of such reactive serine. And we see in the first place that glycopeptide transpeptidase comes to bind onto one of the peptides. And take note that you are having an interaction between the serine residue in the active center, that's reactive serine, that was shown up there, and the amino acid in that chain is alanine. So you have the keys over here, all those colors. The green spheres refer to lysine, blue glutamate, and the red is alanine. So you see alanine is what enters into that active center. And mind you, the active center, you see the active center is like a chair. That active center is having like some two pockets, one short one long one, and you have the peptide chains coming in. So if you have a second peptide that is coming in there from another glycopeptide, you see again it is alanine, which is at the end of that peptide chain, which is getting into that long pocket of the enzyme. And what you see is that there is yellow linkage, the peptide linkage, what's referred to as the pentaglycine chain, because you are having a cross linkage between alanine and lysine. So that's a linkage which is formed. So the enzyme is responsible for the formation of that cross linkage between the peptide chains that are on N-acetylmuramic acid. That's a function of the peptidoglycan. The function of the glycopeptide transpeptidase in this process of the peptidoglycan biosynthesis. All right. Now, of course, the last part, it means that the bond is formed, the enzyme dissociates and moves away. So, I mean, from top down, it's numbered 1, 2, 3, 4, 5 for you to see the steps by which this, formation of the cross linkage between the peptides is formed. Now, you see, penicillin is now being shown. You know, this orange, sort of as if it is 6 or 9. Let's say it's 9. Being shown as penicillin, which comes into, the process to inhibit the enzyme. The first part we have already seen, which is how the enzyme helps to form the cross linkage. Now we see that penicillin, which is being shown as this yellow, or is it orange? In the beginning, it's having some circular area, but that circular area, when it has interaction with that reactive serine of the glycopeptide transpeptidase, what is also referred to as a penicillin binding protein, then the ring opens up and you have penicillin binding into the active center of the enzyme. And once it binds into the active center of the enzyme, the enzyme becomes inactive and so can no longer carry out the formation of those peptide linkages between the glycopeptides. In that way, it means that penicillin inhibits the formation of the peptidoglycan so that the cell wall is not being formed again for the bacteria. In other words, penicillin, slows down, inhibits the growth of bacteria. So penicillin is a bacteriostatic antibiotic. Now we see here, we are seeing the structure of penicillin being compared to the structure of the glycopeptide, which binds in the active center. And these portions are just showing like the alanine residues, which are binding in both the long and the short, pockets. You remember you have alanine at the end of the peptides, and those alanines are what enter into both the short and the long pockets of the enzyme active center. And so these portions, so you see, these portions are opened which have that interaction with the active center of the enzyme. If you look at penicillin, you see penicillin is having a some cyclic structure before. When it interacts with the active center, it opens. So you see you are having that ring in that structure where you have like a 4-sided ring fused with a 5-sided ring together to show that's actually the active component of the penicillin family of antibiotics. And we see that activity, we can look at it in the sense that if this is the glycopeptide transpeptidase and the active center is being shown here, serine, that reactive serine, OH on it, penicillin comes to bind onto that serine, the reactive serine, to form a penicillyl enzyme complex and renders the enzyme inactive. So that's summary of what actually is happening, that you are having penicillin binding to the enzyme and then it just makes it inactive. And that ring which you saw is what is being referred to as the beta-lactam and the beta-lactam ring. So you hear a lot about this. I think one of these pharmaceutical companies in Ghana, I think all of them, those who produce these antibiotics, like is it Entrance Pharmaceuticals or things like that. I'm sure if you go to their website, for instance, they will be having a lot of these things writing about. You can go onto the website of some of these pharmaceutical companies and see. Maybe I'm sure those who are producing some of these antibiotics will be talking a lot about maybe beta-lactam antibiotics, and this is what is being referred to. The ring that you see in penicillin, you have a 4-sided ring fused to the 5-sided ring like that. That is, or even here you have a 6-sided ring. That's what is being referred to as the beta-lactam ring. You find it in this class of antibiotics, just like penicillin, being referred to as the beta-lactam antibiotics. Sometimes they refer to them as suicide inhibitors. It's like the ring is closed, they are inactive, but when they enter into the system and they interact with that glycopeptide transpeptidase, then the ring opens and then binds to the active center and then renders the enzyme inactive. You have these, these different types of rings which you can find. Of course, not exhaustive. There are more that you find different ones. I think the common antibiotics which you find in Ghana in this class are amoxicillin. You see the structure of amoxicillin is having that ring in it. This ring should be, I think, the first one, which is pinam. So that's a pinam. Amoxicillin is a pinam. It's having a pinam ring there. And you also have clavulanic acid. I think you have it as I think on the Ghanaian market now, we have amoxiclav. So you have a combination of amoxicillin and clavulanic acid. And even this ring in clavulanic acid is not here. It's not up here. Because what you see is the same. Is similar to pinam, except that in place of the sulfur in that 5-sided ring, you have oxygen. And because of that, that's an oxapinam. It's an oxapinam, clavulanic acid. And so you have a combination of amoxicillin and clavulanic acid, which is referred to as amoxiclav. All right. Now we can take something on painkillers, like non-steroidal anti-inflammatory drugs. So we have the synthesis of prostaglandins. Don't pay attention to all the things over here. Just let's look at just from the top to prostaglandin, just the first two arrows. So you have diacylglycerol phospholipid, undergoing some conversion into arachidonic acid. Arachidonic acid, we're having 20 carbons in it. So if you're having a diacylglycerol phospholipid, it means you are just joining them phospholipase. These enzymes are joining these diacylglycerols and to form arachidonic acid. And on the basis of this arachidonic acid, you have these enzymes, cyclooxygenases, which convert that into prostaglandins. And prostaglandins are these lipids that, they have some kind of hormone-like effects controlling pain. They actually, let's call them, they are responsible for pain recognition, for instance. So, of course, some of them who are involved in vasodilation. They rather open blood vessels. Some of them will do the opposite, like they go into blood clotting, like the thromboxanes. So you see the thromboxanes on them, you are having a platelets, and then you can have some blood clotting effects occurring, whilst you can have a prostacyclin will be doing the opposites, acting as a vasodilator in that process. So they are involved in like pain regulation. So the inhibition of production of these prostaglandins will result into pain relief. Now you see the enzymes that are involved there, COX-1, COX-2, that's what is referred to as cyclooxygenase. So these are cyclooxygenases. I think maybe some of you have been working in hospitals, some of you in pharmaceutical industry, or maybe in the pharmacy. You might have seen some medicines like that they've written, maybe COX-1, COX-2, something. Even, I think some medicines may even be having that name, COX-1, COX-2. Or maybe if you look inside a leaflet, it's a painkiller, and when you are looking at the pharmacokinetics, they talk about COX-1, COX-2, cyclooxygenases. All right, so let's zoom in on that on that reaction where you have, the formation of prostaglandins. You have brufen. It's a common non-steroidal anti-inflammatory drug, which is an inhibitor, to cyclooxygenase. So this is cyclooxygenases, COX-1, COX-2. So these are responsible, for the conversion of arachidonic acid to prostaglandins. Arachidonic acid is a fatty acid, 20 carbons, and to form a ring structure in that to give you the structure of the prostaglandin, the cyclooxygenases act, and brufen goes in there to, inhibit the activity of these COX, cyclooxygenases, and thus the production of the prostaglandin. In that case, the effects will be some pain relief. I think we'll take a last one, which is aspirin. So aspirin is also doing a similar thing. So you see arachidonic acid, conversion of it into prostaglandin. Aspirin is inhibiting cyclooxygenase 1. So once you don't have prostaglandins that are being produced, the result is going to be pain relief. So, well, I think, okay, maybe this should be the last. Irreversible inhibition by diisopropyl phosphate. So this is, if you take some pesticides that are in the group of organophosphorus pesticides or organofluorophosphorus pesticides that act as inhibitors to the enzyme acetylcholine esterase. We saw that already, involved in the contraction relaxation of muscles. So what it means is that it's actually acting on the nervous system. So what it means is that this DIFP will be inhibiting the enzyme acetylcholine esterase, but in this case, it binds covalently to the enzyme. So if this is the whole enzyme in the active center, again, you see serine. And you see that DIFP binding, on that serine in the active center. So you see that enzyme inhibitor complex being formed. What it means is that acetylcholine esterase becomes inactive, will no longer be acting in that process of, acetylcholine sending the signal, because it's a neurotransmitter to the muscle, and then there's contraction. Then later you have the acetylcholine esterase that will hydrolyze off the acetylcholine, and then another signal comes. That will no longer happen. And what it means is that this DIFP then causes paralysis. So there's paralysis, there's no movement of muscles. The central nervous system is no signal, is coming, is broken down. In the end, that paralysis will result into the death of the insect. So of course, even though the pesticide is meant for an insect, I must add that some of these pesticides actually can also have adverse effects on humans as well. Sometimes, maybe not immediately, but accumulatively. A lot of these pesticides, if you pay attention, they are lipids. They are lipophilic. That's the reason why you see, for instance, you see those ones that the farmers use. You see when they mix them, it's like it foams. It foams because there is an addition of some surfactant in there, because it's oil, and in order for it to mix with water, they have to add some kind of a surfactant in there. In fact, some people even add some little liquid soap in there to make it soluble in water before they spray it. And because it's lipophilic, over time, when you are too much exposed to these things, you can have accumulation of these things in the adipose tissue to get stored under the skin. And it's as if it's not there, nothing is happening. But later, maybe in old age, or maybe in some other disease condition, that you start to lose weight and they get released into the bloodstream. That is when, the effect starts to come out. So in fact, it's of great concern that you have indiscriminate use of some of these pesticides, herbicides by farmers, uneducated, doesn't understand anything about chemistry. Some of them actually even think that they want the substance to be more effective, and so they don't even go according. After all, you can't even read the dosage that is there, and so they want to just add more and more, and it's all over the place. Actually, those things should be handled by some trained officials. Unfortunately, that is not the case in our place. And you must also be careful. I mean, just going and buying some insecticide and start spraying at home all over the place, particularly when you have old people around. I mean, you should be concerned about some of these things. All right, so that is a practical aspect of some of these things, not just learning to pass exams, but to practice it in our lives. Okay, that brings me to keep quiet and now listen to you. So the floor is open. If you have anything to say, you can write in the chat. You can raise your hand. I will enable your microphone and you unmute yourself and then you speak. Then I can respond. So the floor is open.